Each year, Microsoft Research hosts hundreds of influential speakers from around the world, including leading scientists, renowned experts in technology, book authors, and leading academics, and makes videos of these lectures freely available.
Good afternoon. We're going to start the last section of this day. The name of the section is Perspectives on Health Intelligence. We're going to introduce three speakers, but by the first time, I would like to introduce myself. My name is Leila Pontes. I'm representing Gerson Lima Silva, the Scientific Director of Carlos Chagas Foundation, supporting science, technology, innovation in Rio de Janeiro. I'd like to thank you very much, Microsoft, and their organizers to invite me to be a chair of this section. It's an honor for us and a compliment to work with you to establish a very good technological site for developing TI in Rio de Janeiro. I'm also a teacher of a Federal Institute of Rio de Janeiro that work with several kinds of fields in technology also. We're going to start the lecture, introduce Dr. José Cláudio Terra. Each speaker is going to have 15 minutes. They're going to conduce a lecture, and by the end of the 3 lectures, we're going to have 15 minutes for questions. And I'm going to introduce the next speaker, Marcel Jakowski. The presentation title is Impact of Biomedical Imaging on Healthcare. I'm talking a little bit about his bio. Dr. Jakowski is an assistant professor in the Department of Computer Science at the University of São Paulo and manages the Medical Imaging Group. Prior to that, he was postdoctoral fellow and held a research scientific position at Yale University in the Department of Diagnosis Imaging. His research is oriented towards developing scalable biomedical image analysis methods. He has been principal investigator of several FAPESP and CNPq grants. Collaborates actively with Atinola A. Martino Center for Biomedical Imaging. Thank you very much.
Thank you for the introduction. Well, I'm glad to be here today. It's actually my second time in the Microsoft event. It's probably been about 4 years since my first attendance. And I'm very glad to see all the technologies that have been portrayed throughout this event, throughout these 2 days. Today in particular, I would like to share with you actually what we've been doing with imaging and medical imaging specifically. I've seen some of the imaging technology or image applications that have been, under works with Microsoft. I know there's a whole sector on biomedical imaging up in the US. And just like to disclose some of the stuff that we do and show what are the challenges that we have to deal with when we deal with in the medical domain, rather just looking at images in which we deal with visible light. As we know, all these biomedical imaging techniques, such as ultrasound, CT, MRI, PET, SPECT scans, histology for studying pathology, all these things have become really powerful diagnostic tools for all doctors. Okay? It helped not only for diagnosis, but also for therapeutical protocols. Now, as you can imagine, anyone that actually goes to a hospital to undergo one of such exams, well, of course you're worrying about what's going on with your body, okay? That's why you're going in there. All these images are actually stored in PACS system or databases, which are database systems of images in all these imaging centers. And a major concern is that nothing is actually performed, or no analysis is actually performed on those images after the patient leaves the hospital. So the big question is that that's something that we may want to discuss here in the panel afterwards is there something useful that can be done with it? I mean, is there some statistic that can actually be achieved from analyzing all these images that are there? And one of the key challenges is these are all completely different nature of images. I mean, they come from different physical properties. You're measuring different physical processes. Okay? Therefore, we believe that biomedical imaging, in combination with the clinical history of the patient, and as well as all the molecular and proteomics and genomics, can actually increase the knowledge about a population's health, and probably benefit healthcare policies in the future. So there's a huge, as Claudia was telling us about, there's roughly 1 million of images or 1 million exams that are conducted just at Einstein. And you can imagine how many different modalities, what is the percentage of all these that are actually split in this million or so. And this is within just one institution. Can you imagine in all the centers around the world, or even here in Rio or São Paulo, okay, which concentrates all the major medical institutions here in Brazil. So this amount of information will actually overwhelm individual machines, as doctors used to use their own laptops and workstations back 15 years ago, which now is completely unrealistic if you really want to perform large-scale statistic analysis on patient databases. So another key point is each one of these images, they are generated by different probes, different imaging devices, and each of them actually generate a signature. So they are different in terms of their nature and in terms of the noise characteristics. And this is another challenge when we try to devise machine learning algorithms or even statistical analysis tools to be able to cope with all these. Nevertheless, what we want is to continuously track information so that we can quantify it and perhaps share with our peers, especially with other doctors around the world, the conclusions that you get from your information that is sitting there. Okay? And ultimately, this is how we're going to improve health, right? By sharing all this knowledge that has actually been scattered in all those databases that are just remain unused up to now. So what I would like to show you from this point on is 3 applications that have been developed or under development with our group back at University of São Paulo. And these are student projects. These are master's and PhD projects. And to show you how high-end can an application in medical imaging be. So the first example that I would like to show you is on cardiovascular disease. And you see 2 images there. On the left-hand side, you see what we call the circle Willis. And the circle Willis is a sort of a vessel architecture that is actually located in the back of your brain and helps actually communicate all the blood vessels throughout the other places around your cortex and so on. As you see on the right-hand side, there's a big pouch in another patient, as is shown by the yellow arrow. And this is what an aneurysm is. So it's this little pouch that develops, regardless of their cause. It may rupture because of the bigger elasticity that is associated with the walls. And if it ruptures, we are in a big, big trouble. Now, how can we help, for example, detect this in an automated manner? It's just not about looking at the pixels themselves. You need some prior knowledge to understand that this is actually a bad thing. So in order to do these things, this kind of processes, you need to learn what the doctors do, what they have actually learned from their schooling. Okay? And then we can mimic the cognitive system of the doctors themselves to be able to put in those high-end applications. So in this case, what I would like to show you is that we can actually reconstruct vessels and characterize vessel problems, synthesizing angiograms. Why? We want to try to build synthetic vessel architectures so that we are able to create a model from which we can actually get the answers we're looking for. All right? And this is sort of mimicking the nature or how the angiogenesis actually take place. So what you see here on this slide is a vessel architecture on the far left-hand side, which gets processed and then represented as a grammar. So an entire vessel architecture can be represented by a string. And what we call this a sequencing. We call it the sequencing, like those were because of the genetics connotation to it. And so if you represent these as an expression, we can not only encode the information of the structure of the vessels, but also be able to infer and make questions, ask queries, do queries on the string to see how this is different from normal. Okay? And we can learn different grammars according to different vessel architectures that we want to probe for. And here I show you some examples of the vessels that can be easily accomplished or created synthetically. These are angiograms, artificial angiograms. And all the vessels that you see here, they represent pieces of different architecture within different organs. And as you can see in the figure J, which is at the bottom line, you see those little red arrows, they actually show two anomalies or abnormalities. And one of them is actually an aneurysm. So if we're able to create this effect in an artificial image, which can be corroborated with the real images, then we can ask the question, what kind of features from this then can characterize an aneurysm? And then help diagnose this and help the doctor easily locate this, because this patient doesn't really have much time until he really needs to get operated upon. So I'm going to switch gears to a different application, and another one is sepsis. A lot of, as Claudia was saying, only a few doctors like to wash their hands. Okay? So in effect, what happens is that there is when you go into the hospital and you undertake some kind of procedure, and it is very likely that you may end up with bacterial infection, which causes inflammation and basically create a systemic inflammatory response all over your body. And this can easily leads to death. Now the question is, how can you detect this early enough so that you can actually take the proper measures and counterattack this? Okay? And to be able to actually do this, you need to look at the very fine scale of what's happening within your capillary system. So with the same idea, we can use optical imaging, which takes videos at very microscopic scale of the vessels, and you can actually see the blood cells streaming through all these capillaries. And throughout time, you can see that as bacteria starts to spread out, tissue becomes inflamed, and the vessels simply cannot supply the oxygen anymore. Okay? Now, what is cool about this? Well, we can record a video of this in real time, and then we can apply all our computer vision and basically all the machine learning techniques that we've come to learn so far to diagnose this. Okay? And this is exactly what you've done. With simple support vector machines, you're able to categorize different levels of sepsis, of commitment of sepsis, given a certain patient. Cancer is another big topic. Of course, we don't know the exact mechanism of this. I'm not here to tell you any about that, but we know that a lot of things modulates, you see the development and actually how it happens in the human body, such as life factors and environmental exposure. There's one particular kind of tumor, or cancer, which is called a glioblastoma, and glioblastomas are very malignant tumors that occur in the brain, okay? And they're due to, that arise from a glia, or specific cells that actually make up the supportive tissue of the brain. They represent about 15% of all these tumors, and are, they spread out very quickly. So what we want to do here is be able to at least give a sense for the characterization of how this tissue is actually being affected. How can I actually tell that a tissue is affected by this tumor, it has tumorous cells or not? And more recently, there has been a greater advance in all the imaging techniques that we see. Okay? And one particular advance is what is called diffusion imaging, which is an MRI modality that can actually probe for tissue integrity. And I'm going to save you from the complex details because it actually records proton diffusion in ways to infer the integrity of tissues. Now, this has been readily used to, I'm sorry about that, let me go back to that one, to be able to characterize tissue versus normal tissue versus tissue with some sort of an abnormality. In this case, what you see on your left-hand side is a typical intensity-based image that really doesn't really tell much except there's a big blob, which is definitely something that is abnormal when you look at an image like that. On the right-hand side, though, you have a model, you have an image that tells much more information about what's going on inside. And all these little red and blue and purplish glyphs, which looks like balls or spheres, but actually are supertoroids. Okay? And this is at the same time is a geometric model that has been used to represent the information from this, this modality of imaging by reducing the dimensionality. This is something that we are accustomed to. And at the same time, I can actually probe for discriminative information. And now I can actually tell what is edema from what is dead tissue, from what is normal tissue. And this is within reach. Okay, it's just we have the tools, we have the expertise and the manpower to accomplish things like that. These are very complex stuff. So just to show you how bad this thing is, on the top line, on the top row, you actually see maps of a normal brain. And on the bottom one, you see maps of a person that has actually died of it, died of brain tumor. Okay? And the key thing is to learn how this process is, how this proliferation goes on, and how this actually tissue is impacted by this. Here's the same similar picture showing what's happening to the wiring of the brain as a result of this mass expansion. So it's not because we have tumors or bad cells that reproduce in an uncontrollable way, but also we are confined to the skull, to the brain, and therefore everything gets pushed. So this constitutes 3 different applications that we can invest our efforts on. This is just 3. There are thousands of applications that are there just waiting to be discovered, and in order to benefit, you know, our healthcare system and not only health in general. So I'm thinking about, I'm a little too early today, but what are my conclusions here? To be able to work with medical imaging, you actually need to think outside the box. So you cannot simply go through your machine learning algorithms or your statistical data analysis and think that medical images are simply made of pixels. You need to understand what a pixel is in terms of a physical property. That's the first thing. Second thing, you need somehow to approach doctors. You need to learn a bit about anatomy, about physiology, to be able to understand a little bit about human body. Third point, you need to understand the imaging techniques. So it's not like we always take for granted when we see a digital picture made from a cell phone camera or a professional camera that has a CCD. There's some noise patterns associated with it, but we never really question. We assume that the image is perfect, and that's it. I start from that. Here, no. You need to understand what are the noise characteristics of your device and what kind of information is a give. This is very important so that we can actually get the features that represent what we're trying to assess. And more recently, a lot of new techniques have been developed. Okay, so I've been in touch with several groups around the world, and they constantly come with new imaging devices. And these imaging devices now produce terabytes of information. It's not like a 2-dimensional image or a 3-dimensional image. Now we're talking about half a terabyte per patient. Now multiply this by 1 million. If you want to do a cross-group analysis with all the database of information that you have hanging in all these hospitals or imaging centers, you need to be able to cope with this. Okay? And it's very rare that hospitals or medical centers have this IT power to accomplish such a thing. Okay? Probably Einstein is a rare example here in Brazil. This is what I would like for it to happen. I would like this data to be shared, and we can always invest ourselves into building some kind of platform that will allow not only the sharing of information for doctors, an exchange of information between doctors and be able to tell, create new knowledge or devise new therapeutics or new diagnostic tools based on the information in a way that actually will make sense and improve human health. And I've seen that cloud computing is such a buzzword today, and this is something that is here for us to stay and is actually being used for this. There are a bunch of efforts around the world in trying to create such a platform to be able to analyze statistically all these images. But so far, I haven't seen any major hospital or medical institution that actually embraced such a platform. And I think it's an opportunity that we would like probably to discuss. I think this is something that's worth going. And last is that the integration of imaging plus the clinical history of the patient plus demographics, okay, can tell a lot about what's going on with your population. And this is completely disregarded. And this is something that, I'm willing to work on, and I'm willing to share, what we have been doing in regards to that with all of you. So I thank you. Thank you very much for the opportunity.
Thank you very much, Dr. Jakowski, for your presentation. And I'm going to the next introduce Michael Ziskowski. Demo presentation, Project Premonition. Presentation title, Project Premonition, Preventive Monitoring of Infection Agents. Michael Ziskowski manages a cross-discipline team of engineers in the development of research technology into scalable working solutions. He works with academic, industry, and government NGO collaborators to build partnerships and community ecosystems. Recently, he has been focused on projects related to the confluence of aerospace and computer science and engineering, with projects like window flow, Premonition, and the Red Bull Air Races. He also leads research news, online news aggregation service for the academic research community.
Very good, thank you. Thank you very much for that introduction. It's a pleasure to be here. I'm very honored to be asked to be a part of this panel, and I was really glad to see that Claudio didn't make a Pollock joke, because he would have got 2 for 1 on this panel, and I probably would have had to buy him a drink later tonight. So I'm here to talk about Premonition, and this is a really exciting project, cross-discipline with lots of challenges, very ambitious, and I'm representing a much larger team than just myself. There are a lot of smart people on this project, and I thought I'd just quickly go over the people in Microsoft Research that are working on this and some of their disciplines, and then also talk about our collaboration with academia. We have people on this slide from the programming languages area of research, artificial intelligence and machine learning, computational genomics. We actually have a few people on staff in that area, the hardware engineering, and then research engineering. And there's a lot of collaboration that has to happen inside Microsoft Research to pull off a project like this. And then outside the core sort of computer science research areas, we've partnered with a lot of other academic institutions that specialize in different areas of the domains that we're exploring. We've also been fortunate enough to have joint funding from the Intelligence Advanced Research Program Association, IARPA, who is helping co-fund these universities. So it's really stretching our investment dollars as well. Vanderbilt, Penn, Pittsburgh, UC Riverside, who has some of the most advanced mosquito analysis people on their staff, University of Kansas, and then Johns Hopkins University, and Doug Norris here is a world-renowned mosquito expert. So I'm really excited to be here. I know this is an interesting topic for many parts of the world, but very interested in also connecting with people in Latin America around potential future collaborations. So if anything you see here looks like something you might be interested in getting involved with, please come find me. Okay, so, oops. So of course, catastrophic diseases are very impactful to humanity, not only in the loss of life or impact to health, but it's very expensive. And even if the disease does not become an epidemic, they're very clearly very impactful around the world. And this is just an example of the different types of infectious diseases that we believe could see some improvement in an early warning system. And that's essentially what we're trying to build, is an early warning system. The original idea for Premonition was somewhat driven by malaria and then Ebola. That was back in 2014. We also have done exploration around chikungunya, and I'll talk a little bit more in detail about a field experiment we've attempted there. But of course, Zika is the next big thing. It doesn't seem like there's any lack of opportunity to do better in this space. And we're continuing to evolve our thinking and our exploration of how we can help these different early warning systems for these different diseases. So I'm going to baseline here just on a couple things before I drill into the project. You know, the over 60% of disease emergent events are passed from animals to humans. And this is a really important aspect because there's lots of things happening out there in the environment, especially with animals, that aren't really a problem until they get into the human population. And the interesting thing about that is to the current state of the art and to predict when that's going to happen or the impact of how that transition happens, called zoosis. It takes about 2 weeks for the World Health Organization to actually come up with a declaration that there is an outbreak of something that we need to do something about. And that's a long time. And we believe we can implement a system that will help us actually see what's happening in the environment ahead of time so we can be much better prepared and try to contain these things. The other interesting stat that we've come up with is that when there are pathogens found in the environment, it's believed that over 70% of those are really unknown in the scientific community today. And that's kind of a scary statistic because we don't necessarily even know what's out there that can cause us problems. And so the more, even if we find things in the environment that aren't necessarily a problem yet, we can be better prepared and better understand the environmental effects of these things. Okay, so I guess I can look down here. So we need better environmental surveillance. And we want to see what's circulating in the animals before they impact humans. A little bit of a spoiler alert. We want to build an autonomous, large-scale, self-adapting cyber-physical system. I like that term. Really, it's something that has the smarts and the capability to go out in the world and collect information for us that help us make better decisions. So there's 3 key innovative tracks as part of this project. It's very ambitious, and all 3 of these things are very challenging. The first is that we want to build a smarter mosquito trap. We believe mosquitoes are a sensor in the world that we can take advantage of. They go around naturally, collect blood samples for us, and then can return those blood samples to us via this process. Existing mosquito traps are very low throughput. They must be hand placed by experts and require a lot of manual processing. So that's one of the technology areas that we're working on, and if you came to DemoFest, I have our prototype with me. The other one is the advancement of drone technology, and there's so much happening in this space. It's really exciting. There are some really hard challenges there around safe, autonomous operation in unknown environments. And we are working on a stack of technologies that help us make sure that the system is reliable and verified and secure, up through controllable to all the way to the high-level decision-making so that these things can go out and deploy these traps in smart ways. They can deploy them in an area where we believe there's going to be mosquitoes, and then we can go retrieve them to retrieve the mosquitoes. And finally, the third piece is the metagenomics. As we collect more information from the environment, we need to scale up our ability to process this information. And it's kind of what Marcel alluded to in his talk. This is a really important area. We want to sequence the DNA from the mosquito and evaluate what its blood characteristics and the protein characteristics, what pathogens it's carrying, and what it may have actually bitten. We'll be done on Friday. Okay, so I mentioned mosquito as a device. They grow naturally in the environment. They don't select certain homesteads. They're pretty ubiquitous. They like to bite lots of things, And so we can get a broad scale understanding of what's happening in the animal world. This actually isn't a new idea. There are classical entomological methods that are used to collect mosquitoes, but they're, as I mentioned earlier, they're very cumbersome, and this is something that we can leverage to help our understanding of how to make it better. So this is a picture from the island of Grenada. We actually did a field experiment. We kind of did a baseline to see, okay, is this project really crazy? Can we actually pull this off? What do we think we would need to do? This is the state-of-the-art mosquito trap. And it is literally a little igloo cooler where some dry ice is put into it. The dry ice emits carbon dioxide, which is a primary lure for mosquitoes. That's why mosquitoes bite you. You breathe out carbon dioxide, they go, ah, lunch. The rest of the trap hanging below that is a net, and it includes a ultraviolet light. Mosquitoes are also attracted to ultraviolet light. It's attached to a big, heavy battery at the bottom. The mosquitoes and a bunch of other stuff fly into this netted trap and end up in the bottom of this container. And they're typically left out, 18 hours. They're placed, and then the field biologist will, and placed by hand, very heavy, very cumbersome, and then we'll come back 18 hours later to retrieve it. Hopefully the local teenagers didn't do something weird to it, like treat it like a piñata. This is Doug Norris. I mentioned him earlier. This is what comes back. This is the bug goo that you get out of the trap. And it takes somebody like this to then separate the mosquitoes out of the bug goo. And this is literally the state-of-the-art process that's used today. You can imagine how time-consuming and expensive this process is to get an understanding of what's happening in the environment. So this is our vision for what the trap should be able to do. We're still evolving our thinking. We're working on different prototypes, but this is really the high level. We want to do selective trapping. So if we can just trap a mosquito, or even just the Aedes aegypti mosquito, then we don't have to do any separation. We have the mosquito we want. We have the sensor information we want. We want it to be low power, low cost, so these are easy and cheap to make, no moving parts. We want to make these available to the world. This isn't really a business that Microsoft is looking to get into. We're really thinking about this as something that we can work on together to help solve this problem for humanity. In situ stabilization, the mosquito needs to stay alive for as long as possible. Otherwise, its DNA and blood structures start breaking down and we don't get as much information out of it, or it becomes unreliable. And we want, I guess we need low-cost emitters, different sensors to determine if it is a mosquito that's entering in the trap. So this is where we're at today. This is, if you came to the demo fest, this is what I was showing. It mostly works. This was a very early prototype. This is very early stages of the project, but we've had really good success with this device. This is completely custom designed and built in-house in MSR. It has about 68 cells, and they are designed to capture one mosquito. It's heavy, but it works. We've tested it in a laboratory environment. It works by looking at the signal of mosquitoes. And these are, the top line are what's a measured wing frequency beat of the mosquitoes, and the bottom are the classifiers we built, and we use those with the IR sensor in the trap to determine whether or not it's a mosquito. And we had over 90% success rates with this trap in a laboratory environment. One of the interesting surprises that happened for us is, as we were running this experiment, we noticed by collecting the semantic metadata around when these mosquitoes were entering the trap, we could find patterns in their behaviors that weren't previously documented. There had been some studies in entomology around how mosquitoes behaved and how different mosquitoes behaved. this chart on the bottom is the Aedes aegypti, and it has a much more aggressive behavior than the mosquito on the left, which has a more circadian rhythm. And this will help us in understanding better how to place the traps and maybe how to be more effective in our capturing of these bugs. I'm going to have to start moving faster, but I did want to show this. This is actually our laboratory experiment, and this is Ethan Jackson, one of our leading researchers on the project. And if the video, I don't know if I can get that video to play. Anyway, this is in the UC Riverside laboratory where we were able to get such good results with the trap. Okay, so that's the trap, and that's the trap challenge. We also need to solve the drone challenge. And there are a lot of people working on this. There's a lot of universities working on this. There's a lot of industry people working on this. We've currently broken this down into 4 components. As I mentioned earlier, that secure OS layer, the sensing layer, making sure we can control the device, and then the high-level planning. Ultimately, from the reliable platform all the way to decision-making under uncertainty. I'm going to go through these pretty quickly because I know I'm running over. The idea on the secure OS side is that we're leveraging tools already being worked on and delivered from Microsoft Research. Z3 and Boogie are already available. Verve OS is another one we're building out. And the idea is that this is a fully verified and functionally correct system that when it's deployed, we have a high confidence level that it's going to work correctly. In the sensing space, we have some of the smartest researchers in computer vision, machine learning, and some of the best tools in the industry that we can leverage to help in understanding the environment as the drone's out there and sensing the environment, understanding what it's seeing, and providing classification of that environment information up to the next level so that then decisions can be made. In the correct control space, our focus here is really on the degradation of signals and information. If everything's working perfectly, it's pretty easy to fly a drone around. If you start losing information or things become unreliable or maybe you lose a signal, that's where things get interesting. And we're really excited about some of our work here, and we have a paper coming out in a robotics conference in a couple months. And then finally, the high-level planning, and this is really where the artificial intelligence component comes in. How do you know where to place the traps? How do you know to avoid this area or that you want to go to this area? And there's a lot that goes into this. This is a very rich area of research right now, and we're actively studying that too. Okay. So that really was me out there. I am a drone pilot. I'm actually a real aircraft pilot, and I'm an aerospace engineer. So my specialty is really in that domain. But like Rico mentioned at the opening of this event, it's really important to work across domains as well. And that's really what's making this project work, is that people are learning each other's languages. We're combining computer science. We're pulling together the different domains, and that's when the magic happens. It's when you get these hard problems together and people from different domains working together and understanding each other's languages, understanding each other's problems, and then coming up with solutions that can really change the world. I'm just going to, maybe I'll talk about this quickly. This is the third trajectory. This is around the genomics problem. You have the problem of the host and vector, the microbes inside the blood, and the proteins. There's a lot of people on this project working on this problem, but especially as it relates to the healthcare space, this is a great area for us to collaborate on. Eventually, this is what we want to get to. We want to improve our understanding. This is from healthmap.org, and the numbers are pretty small on this chart. We want to increase this by at least an order of magnitude and at least 10 orders of magnitude in terms of frequency of understanding. And that's about it. Thank you very much.
Thank you, Mike. I'd like to thank our 3 speakers for the outstanding lectures on health intelligence. And you can start for questions. First, I'm going to make the first question be mine, okay? And we know that Brazil is a challenge for health intelligence and healthcare, and for the difficulty about as a huge country. How you 3 think we can overcome this problem as soon as we can, using health intelligence and make translation medicine available for all the population, even for the poor and in the distant places in Brazil?
Hello. I think that we have evolved a long way from a decade or so, in a way that we're able now to have doctors in really remote regions and being able to relay their exams or medical reports back to big centers and be able to actually get a second analysis, and if they're unsure what they're seeing, and get back to the to those remote regions back, and in a way trying to enhance what's happening. Another point that I'd like to make is also I hope that the government is also have to have this conscience of trying to spread out these efforts throughout the country more evenly. Okay, and I think institutes like Einstein and different hospitals are actually doing the same effort and trying to create small centers around the country, and this will disseminate not only the experience from the doctors, but also the technology that has been developed so far. So I think we're on a good direction.
I don't know if I could improve on either one of those. That was great. I totally agree. I think collection of data, the big data, paradigm shift is here, and with that comes, the machine learning technologies and AI, and allows us to do a lot more things with all of that information. So, fundamental to that, though, is access to the data, and it can be challenging to share that information. I see that in different industries, and healthcare, I think, is one of those. There's a lot of privacy concerns and regulatory issues, and those can kind of get in the way of knowledge and knowledge sharing. And I really encourage the community to think about ways that we can work around those issues, because I think that could help explode our understanding and knowledge sharing. And then, yeah, experience, sharing the experience. I totally agree with that. I also would maybe just add that it's that collaboration between different domains that I really think is important. And I've seen things go sideways when people don't even really understand the same language, and not necessarily Portuguese versus English, but healthcare versus computer science. You talk about things in different ways, different domains have different ways of thinking about things, and if you can combine those things and learn each other's domain languages, you can really find some interesting, simple solutions to problems. And that's where I've seen the magic happen, so maybe the combination is great.
Thank you very much. Now we can open for audience questions.
Everybody wants to party.
It's a question to Dr. Ziskowski. This solution, this idea of a drone taking the traps for the mosquitoes, usually they have to fly over cities like Rio, or cities like in Africa or in other parts of the world, yes, where they could be victims of people trying to shoot them or to capture them, to sell them. how do you cope with these kinds of problematic issues that could happen to the project?
No, that's a great question. Maybe, I don't know if you heard about the guy that shot one with a shotgun that was flying over his backyard. He thought it was watching his wife sunbathe next to the pool. And I think there's first off, I think there's a really big PR issue around drones. And I understand the concerns around privacy and the right to not be on somebody's camera, but also balancing that with the benefits that these drones can provide. And believe it or not, there's been a lot of studies in the aerospace industry around the safety of unmanned vehicles versus manned vehicles, or peopled vehicles. And the safety and reliability of these automatic systems are actually better than human error normally, even with a very highly trained pilot. So I think that those type of perceptions are something that we need to work on and garner the support of the community at large. Of course, you know, that's not going to solve all the problems, and I think our approach is really around making it as safe as possible, so that if something bad happens, if there's a bad actor and they decide they're going to take it down, that we try to have enough robustness in the system that when things go wrong, that it fails gracefully. And I think that's about as good as we can do by keeping things somewhat inexpensive and simple, and hopefully, you know, people won't see a need to do that for monetary gain. But that's the best answer I have today.
Now I'd like to thank, in the name of Microsoft Research Summit, Cláudio Terra, Marcel Jakovski, and Mike Ziskovski. And I'd like to...